This video will cover 3 different types of experiments that were performed in the early 20th century that ultimately revealed that DNA, or deoxyribonucleic acid, served as a genetic material in living organisms. The first experiment that attempted to reveal that DNA was the genetic material actually had little to do with DNA at all. Frederick Griffith performed a series of experiments in the early to mid-20th century with 2 strains of bacteria that, at the time, scientists knew quite a bit about. The types of bacteria actually come from a strain of bacteria known as Pneumococcus. You may have heard of pneumonia before, and Pneumococcus is the strain of bacteria that causes pneumonia. And the pneumococcal strain comes in 2 different varieties. There's a smooth strain and a rough strain. Before we get too carried away with the Griffith experiment, I wanted to show you the difference between the smooth bacterial strain of Pneumococcus and the rough bacterial strain. So we'll look at that on the next slide. So what you can see on the screen are 2 very crude representations of bacterial cells. And what I'm going to do is I'm actually going to put a cell wall around each of these cells. Again, very crudely drawn, but I hope you get the idea. Okay. The cell on the left, I'm going to go ahead and make our rough cell, while the cell on the right will be our smooth cell. Okay, so what's the difference between the rough cell and the smooth cell? Well, if you could look at the actual surface of the rough cell's cell wall, you'd see that there are these proteins and carbohydrates that are not found on the smooth cell. In fact, if you could look at this cell under the electron microscope, you'd find that the cell actually appears rough under the microscope, while the smooth cell does not. Well, why do we care about these carbohydrates and proteins on the rough cell that are absent from the smooth cell? Well, it turns out that when these cells come in contact with a host, say a human, or a mouse, or a rat, or a hamster, these proteins on the cell surface of the rough cell are recognized by that host's immune system, and thus the rough cells are targeted for destruction. Okay? The smooth cells, because they lack these proteins and carbohydrates, in essence can exist in the host in stealth mode. The host's immune system cannot target these cells for destruction, and they persist. Because they can persist, they reproduce. And as they reproduce, they have a tendency to make the animal sick. And in some cases, the animal can get so sick that it actually dies. All right, with that in mind, let's look at how Griffith used the rough and smooth pneumococcal cells in his experiments. So, like any good experiment, Griffith used a series of positive and negative controls to determine the significance of his findings. He began his experiment by injecting mice with smooth cells. Remember, these are the cells that lack the carbohydrate and protein molecules on the cell wall surface, and thus can evade the host's immune system and cause the animal to get sick and perhaps even die. And that's exactly what Griffith saw. When he injected the animal with smooth cells, the mice died. So this would have been one of Griffith's positive controls. He didn't stop there, of course, though. He did an experiment with living rough cells. These are the cells that the host's immune system should not be able to ignore and thus destroy. And when he did that, the mice survived, and this would have made the experiment one of Griffith's negative controls. Now in class, we tend to use an experimental design protocol where we have 1 positive and 1 negative control. But scientists don't always stop there. Sometimes they have multiple positive and multiple negative controls, and Griffith did exactly that with his experiment. He had another negative control. In this case, he took the smooth cells that he knew should have killed the mice, but first killed those cells with heat treatment. In essence, he boiled those cells to inactivate any proteins that might have allowed the cells to divide, to reproduce, and really to survive. And when he injected those cells into the mouse remained healthy. This would have been expected, and thus there was no change in the dependent variable, so we can think of this as a negative control. Well, the fourth part of the experiment was perhaps the most important part of the experiment, because here is where Griffith took those heat-killed smooth cells and mixed them with living rough cells. Now you might say to yourself, what would happen if you did that? And that's the question that Griffith asked himself as well. When he did the experiment, the mouse died, and that was curious. Why would cells that didn't kill the mouse, the heat-killed smooth cells here, all of a sudden kill the mouse when they were mixed with living rough cells? Well, Griffith wasn't so sure, so he began to investigate what might have happened. He took a blood sample from this mouse and found living smooth cells inside the mouse. Again, very curious. How is it possible that living rough cells could have somehow been transformed into living smooth cells in the presence of heat-killed smooth cells? Well, Griffith wasn't quite sure what had happened, but what he hypothesized was that there must have been a transforming factor that came from the heat-killed smooth cells that transformed living R cells into living smooth cells. He wasn't sure what this transforming factor was going to be or what it was, but thus the race was on to try to figure out what this factor might have been. So it took almost 2 decades for scientists to produce enough evidence to support the view that DNA was the transforming factor in Griffith's experiments. Griffith didn't know what it was, and it took a long time for scientists to agree that it was most likely DNA. So the question becomes, how did they figure out that it was DNA? Well, it took the work of 3 scientists, Avery, Mcleod, and Mccarty, in the mid-20th century to show that DNA was indeed the most likely biological molecule that was serving as the transforming factor in Griffith's experiments. Well, how did Avery, Mikado, and Mccarty determine that DNA was the transforming factor? Well, it began by taking those living smooth cells that had come from the dead mouse. Remember, I showed you in the previous slide that the dead mouse had living smooth cells present in its bloodstream, and breaking those living smooth cells wide open. And when they did that, the understanding would be that there would be carbohydrates, fats, proteins, and nucleic acids, the 4 major biological molecules that are found in any living cell, now in that extract. Well, Avery, Mcleod, and Mccarty decided to take that extract and treat it with different enzymes that would destroy one of those 4 biological molecules. So, for example, if they treated the extract with enzymes that destroy fat, these would be lipases, the lipid would be gone from the extract. They could take that extract and then see if it was still capable of transforming the living rough cells into living smooth cells. So they did that. They treated the extract with lipases, they destroyed the lipid, they added the extract to living rough cells, and they actually were still able to successfully transform those bacteria. The living rough cells became living smooth cells. So that finding suggested that it wasn't lipid that was serving as a transforming factor. Well, they didn't stop there. They took the extract and then treated it with enzymes that destroyed protein. These are the proteases. When they did that, they took the extract that had been treated with the proteases, mixed it with the living rough cells, and found that, lo and behold, that extract was still capable of transforming the living rough cells into living smooth cells. All right, next, they treated the extract with an enzyme that destroys RNA. We haven't talked a lot about RNA, but RNA is one class of nucleic acid. And there are enzymes that can destroy RNA and not DNA. So Avery, Mcleod, and Mccarty did that. They treated the extract with RNase, destroyed the RNA, combined that extract with living smooth cells, and found that that extract was still capable of transforming the living rough cells into living smooth cells. Well, you can probably see where this is going at this point. When they treated the extract with enzyme that destroyed DNA and not any of the other biological molecules, they found that the transforming activity went away. This was indicative of the fact that DNA and not RNA, not protein, not lipid, and not carbohydrate was serving as the transforming factor. Down below is a kind of a figure summary, which can be confusing if you're not careful. It's going to start by taking the living smooth cells and breaking them open again to produce the extract that would have contained carbohydrate, lipid, protein, and nucleic acid. But then when you see extract from live, smooth, virulent bacteria, carbohydrate, what this is suggesting is that the protease, the lipase, the RNase, and the DNase were all present, so all the biological molecules except the carbohydrate were destroyed. added to the living rough cells to test to see if the carbohydrate by itself was capable of transforming the rough cells into smooth cells. And as we can see here, that didn't happen. So go through these other two possibilities just to make sure that you understand what Avery, Mikado, and Mccarty did to demonstrate that protein was not the transforming factor, but DNA was. The third set of experiments that supported the view that DNA was the genetic material was performed by a duo of scientists named Alfred Hershey and Martha Chase. Notice the date that this experiment was performed in, 1952. You don't need to memorize the date, that's not what's important, but keep in mind that Griffith started his work in 1928. I really should say he published his work in 1928. He started his work before then. And Hershey and Chase published their work in 1952. That's almost a quarter century later. It took a long time for scientists to be convinced that DNA was indeed the genetic material. But onward with the Hershey and Chase experiment. Hershey and Chase took advantage of the fact that viruses and bacteria coexisted together, and that there was a certain type of virus that injected its DNA into a particular type of bacteria. This virus was known as a bacteriophage, specifically a T2 bacteriophage. And as you can see up here, the virus will attach itself to the cell wall and membrane of the bacterial cell and then inject its genetic material into the bacterial cell. But what's important to note is that scientists at the time weren't sure what that genetic material was. Was it protein? Was it DNA? Was it carbohydrate? Was it lipid? They really weren't sure. Well, luckily, viruses are really only composed of two types of biological molecules, protein and nucleic acid. So Hershey and Chase took advantage of that fact and labeled two different populations of viruses with radioactive isotopes that labeled the protein or the nucleic acid specifically. In the first part of the experiment, they labeled the protein coat of the virus with radioactive sulfur. So because viruses are only composed of two types of biological molecules, protein and nucleic acid, it turns out that the outside of the virus is composed of a protein coat, while the inside of the virus is composed of the nucleic acid. So Hershey and Chase labeled the outside of the protein coat with radioactive sulfur. They then allowed the viruses to do their thing. The viruses infected the bacterial cells, and after a few hours, the mixture was collected and put into, of all things, a blender. And it was an actual blender. The blender is still can still be seen at certain museums. But the purpose of blending this mixture was to dislodge the viral particles from the bacterial cell. So Hershey and Chase did that, and then took that mixture and put it into a centrifuge. If you've never seen a centrifuge, I'll be sure to show you in class. But what a centrifuge does is takes a sample and spins it around and separates that mixture into different components. And the components are going to be separated by their density. The most dense materials will be at the bottom of the tube or the container, while the least dense components will be found at the top of the container. So because bacterial cells are more dense than viruses, the bacterial cells were found at the bottom of the container, while the viruses were found at the top of the container. But in addition to that, Hershey and Chase found that the part of the container that contained the viruses also contained the radioactive sulfur. It contained the radioactive protein. If the protein wasn't in the bacterial cells, that made it really hard to support the view that protein was serving as the genetic material, because these bacterial cells had to make new viruses. If the instructions for making new viruses weren't found inside the bacterial cell because they were floating around outside the bacterial cell, made it hard to conclude that protein was serving as the genetic material. Okay, so let's go down below. Similar experiment, but this time Hershey and Chase labeled the nucleic acid, the DNA, with radioactive phosphorus. This is an important point to make. Protein does not contain phosphorus, and DNA does not contain sulfur. So the scientists were able to follow each of these molecules through the experiment by labeling them with two different radioactive isotopes. All right, great. So down below, we've got our experiment just like we had before, but this time the DNA is labeled and the protein coat is not. The viruses do their thing. They inject their genetic material into the bacterial cell. The mixture is moved to a blender where the viruses are dislodged from the bacterial cells. That mixture is centrifuged, separated by density, and this time the radioactivity was found in the pellet, or in the location of the tube where the bacterial cells were located. And this suggested that the radioactively labeled DNA that had been labeled with the radioactive phosphorus must be serving as genetic material because it was in close association with the bacterial cells, while up above, the radioactive phosphorus was not located with the bacterial cells. So what Hershey and Chase were able to conclude is that the genetic material was most likely nucleic acid. It was most likely DNA, while it wasn't going to be protein. So together, Avery, Mikado, Mccarty, and Hershey and Chase were able to demonstrate that DNA was the most likely candidate to serve as the genetic material. These are complicated experiments, and I understand if you have lots of questions. If you have questions, please come and talk to me. I'm more than happy to help you. But these experiments are really important in understanding not only the process of science, but understanding that there were big questions in science that were being asked in the early 20th century, and it took a long time for scientists to answer them. Other scientists performed other work, other data was collected, but these are the two most important experiments that revealed DNA as the genetic material.